have this in your hands because it came out July 2nd, but it was a letter from the National Federation of Federal Employees. And these are the people who make the determinations on toxicity of materials at the EPA headquarters. It's all of the scientists, engineers, toxicologists, statisticians at the EPA headquarters are represented by this union. And this is a letter that they voted unanimously to send out in support of our statewide ballot measure to prohibit the addition of fluoride to the water supply. And I'll just read you one little paragraph here that they put in there. It says, our members review. Remember who these people are. These people that this academy wished that we had on our side. These are all the toxicologists. Our members review of the body of evidence over the last 11 years, including animal and human epidemiological studies, indicate a causal link between. Now, you know, the cigarette smoking people have been fighting over this issue for the last 50 years. They say you can't prove a causal link. Well, here's 1,600 scientists saying that their review indicates that there is a causal link between, and that's human and animal studies, causal link between fluoride and fluoridation and cancer, genetic damage, neurological impairment, and bone pathology. Of particular concerns are recent epidemiological studies linking fluoride exposure to lower IQ in children. Now you can imagine that would have hit with like a real thunderclap across the United States. It was covered by only about 3 newspapers. We sent out a nationwide press release and about 3 newspapers picked it up. The rest of them fell into a coma. But what did happen is that Michael Easley, or some people call Michael Weasley, who is the proponent, kind of on again, he represents himself as either a spokesman for the American Dollar Association or he represents himself as a dental director, I think, of Kentucky at one time, agreed to debate William Hersey on PBS. Now, that's an interesting thing to do. And so what I have here, and everybody, as you walk into the room, come up front and get one of each. One yellow, which is your copy of the letter from the EPA scientists. On the back side is a summary of what they said. America Overdosed is from the Sierra Club of Pennsylvania, where they basically took all the ADA data and the public health data and showed that even if you're a dentist and you think children should have 1 milligram a day, you're getting more than that. And on the other side was our fact sheet. Is basically there's 6 facts. Fluoride causes cancer, hip fracture, dental fluorosis, and dental fluorosis is linked to lower IQ. Large-scale studies show no decrease in tooth decay, and it's not FDA approved. And get this audio tape, because on this audio tape, who you'll hear is Michael Weasley debating Bill Hersey. Who's Bill Hersey? Bill Hersey is the senior vice president of the EPA Scientist Union. He's a toxicologist. And what's interesting is that it's really clear that Dr. Weasley had read Mr. Joseph Goering's description to Hitler on how to be the information manager. Because what Goering said is what you do is you denigrate the source, denigrate the information, and cite authority. And so on the debate, all Michael Weasley said was that, oh, who is this guy, Bill Hersey? He's only the representative of the 1,600 toxicologists at the EPA. None of the janitors agree with him. You know, there's 30,000 people there. You know, so he's just a toxicologist, and the information is, well, it's all flawed. They're confused. They don't really understand the issues. And besides, it's recommended by the American Dental Association. So he did 3 things. He denigrated Bill Hersey, he denigrated the information, and then he cited authority. Listen to the tape and see if you think he won the day, because it was a very interesting tape. Also on there is Paul Connett, who was helpful in our campaign to try to prohibit the addition of rat poison to the water supply of California. And, well, you know, it's not a very good rat poison. Actually, 5 milligrams per kilogram will kill a human. It takes 100 milligrams per kilogram to kill a rat. So, you know, it's not a very good rat poison, but it's really a good people poison. So, anyway, come on up and get your audio tape. There's 100 of them, so be sure and get your audio tape, just 1 per person. And then 1 yellow, one white page. You're welcome to strip my name off of these, reproduce them. If you want them electronically, stop by our webpage and pick them up and put them in your own computer. And so with that, I'll keep it brief.
Thanks, Dave. Thought for a minute we were having a Billy Graham crusade here as they flooded to the altar. Hallelujah! I hope you enjoyed your Mexican lunch that I promised you. It was actually pseudo-Mexican, disguised as delicatessen food. All right. I have the privilege of introducing to you Ron Dressler, who has some comments.
Just for those of you who taking the accreditation exam, room 442 at 5:30. Bring your notebook so that you got something to write on. It's gonna. Right now, I only know of 3 or 4. Unless raise your hands again. Those who plan to take the test. 1, 2, 3, 4, 5. It looks like 5 or 6. Okay. It's my understanding from David Reggiani, it's about an hour and a half to 2-hour test.
Welcome back. I hope you enjoyed your lunch. I want to introduce to you now a man who many of you have become acquainted with his scientific papers. He's a prolific researcher at Wake Forest University School of Medicine. He has a PhD in neurobiology and researches in the area of the physiology and pharmacology departments at that medical school. For 15 years, he's been studying methylmercury and its role in health effects, and more recently, mercury vapor exposure in animals. We're glad to have him, and please pay close attention to him, Dr. Mike Aschner. Thanks.
Thank you, Michael, and I'd like to thank Mike as well for inviting me. It's a pleasure to be here. It's an eye-opener for me in a way. I sat through the morning session. Those of us who are at the bench sometimes lose sight of what's going out there in the real world, and I think it was very helpful to me to see that some of the work that we're doing has some application in the everyday practice that you are involved in. Now, I'm coming from a different background, and I'm going to get even with you for all these x-rays that I've looked at this morning. So hopefully you'll bear with me. Okay, can I have the first slide, please? Okay, left or right? Right. This is right, the forward button. The left. Okay. Okay. No, it's working. Okay. All right. This is where I come from. Some of you will recognize this place. This is Wake Forest University School of Medicine. We just changed our name. Everybody knows about the ACC basketball, and we thought we'd get some more recognition as Wake Forest University rather than Bowman Gray. Actually, I work in this building here, which is downtown Winston-Salem. That's where the Department of Physiology and Pharmacology is housed, and that's where we get all our work done. Now, I want to talk a little bit about our work, and I will start by giving you a brief introduction to the cell system that we work with. These are the astrocytes. Some of you may remember them from dental school. They're beautiful, stellar-shaped cells, and we have the ability to isolate these cells from the brain and grow them in the dish in tissue culture for a few weeks. I will subsequently talk about the metallothioneins, which is an endpoint that we have looked at after mercury vapor exposure, show you some of the data that we've accumulated over the last couple of years, and then I will talk about an enzyme that is an astrocyte-specific enzyme, which is known as glutamine synthetase, and I will give you some recent data. I'll show you some slides about the results from those studies. Now, I noticed everybody starts with a brief history, and I won't be any different, I guess. This guy here, his name is Robert Virchow, a German pathologist, which discovered the neuroglia, of which the astrocytes are a subtype of cells. The neuroglia include the astrocytes, the oligodendrocytes, which are the myelin-producing cells of the brain, and the microglia, which are the equivalents of the peripheral macrophages. And this individual here, who was a neuropathologist, postulated that since the brain can exert its own immune response, that it must have a layer of connective tissue, which he called neuroglia, and that these cells are involved in immune responses of the CNS. It turns out he was completely wrong, but we tend to be very nice in the sciences, and we still credit him with the discovery of the neuroglia. Now, this is from an article that was published in 1989 by Kimmelberg and Nornberg, and I just want to impress upon you the relationship that these cells, the astrocytes, have with, it's a little bit out of focus, with other cells within the CNS. These are the cells that we're working with. They're shown here in orange, and you can see that these cells have extensions from the cell bodies which are known as astrocytic foot processes. Until about 10 or 20 years ago, the dogma was that these cells simply provided cytoskeletal support for the neurons, which are the integrating cells of the CNS, the output essentially of the CNS. But over the last 20 years, we've learned that these cells have a very intimate relationship with many other cell types within the central nervous system. And this schematic just shows you some of these relationships. Over here you have a blood vessel, and you can see that the foot processes of the astrocytes almost completely engulf or surround the capillaries, and we now know the various chemicals that are produced within the astrocyte and the endothelial cells. They can diffuse from one cell to the other. Thus, it is believed that the astrocyte can exert control over the contractility of the endothelial cells that form the blood-brain barrier. And therefore, the astrocyte is very important in terms of maintaining blood flow and regulating blood flow within the central nervous system. Over here you can see the neuron. We talked about this cell, probably the most important cell in the brain, with its extension, the axon. The white stuff here is the myelin, which is produced by the oligodendrocytes. And you can see again foot processes originating from the astrocyte that surround the roads of Ranvier, which are the areas that lack the myelin sheath. And another foot process is shown here around a synapse between two neuronal processes. And we've learned over the years that these cells can mop up, for example, sodium or potassium from the extracellular fluid at the level of the synapse where neurotransmitters are being released. These cells can take up excess of neurotransmitters, and these cells essentially have a whole plethora of neurotransmitter uptake systems and receptors in a very similar way to neurons, and therefore you can view them as a safety valve. Essentially, they are endowed with maintaining the proper environment for the neuron. If there's excess noxious substances on the outside, they mop it up and they can metabolize it or dispose of it or release it back into the bloodstream. This is just another schematic showing some of the transporters that are found on these cells. The focus on this doesn't work. The point of this slide again is to show you that these cells have neurotransmitter uptake systems. I've mentioned that in the previous slide. These are usually sodium coupled, as shown here. They are also very important in terms of maintaining proper pH within the CNS. And they have a bicarbonate chloride exchanger, which is very important in terms of this function. Another very important function is removal, or what's known as spatial potassium buffering. As all of you know, when the neuron fires and the action potential is moving down the axon, potassium is extruded from the cell. And you don't want to have too much potassium in the extracellular fluid because that tends to depolarize the cell and it will repetitively fire. These cells, the astrocytes, have potassium channels, and the potassium that accumulates in the extracellular fluid between these cells can then diffuse by way of a concentration gradient from high potassium concentration into the cell that has a lower potassium concentration. And this mechanism obviously allows for the potassium to move into the cell and into other cells that are coupled to this astrocyte, thus maintaining the proper concentration of potassium in the extracellular fluid. Now, I've summarized, I believe, that's the next slide. Again, I just want to impress upon you the fact that the astrocytes, which are shown again in orange here, and the neuron can be viewed as a unit. There's crosstalk between the astrocyte and between the neuron, and therefore you can imagine that if something goes wrong with the astrocyte, if the astrocyte cannot maintain proper control over the extracellular fluid, that will translate into a dysfunctional neuron. And if the astrocyte per se is dysfunctional, it may release a number of noxious substances such as ammonia, glutamate being an excitotoxic amino acid, quinolinic acid, and many other compounds. So it's critical that this cell, in addition to the neuron, function properly if the nervous system is to function the way it's supposed to. Now, I won't go into too many details with respect to this slide. Just as a way of summary, I've talked a little bit about the function of the astrocytes in terms of neurotransmitter metabolism and modulation. If you recall, I mentioned that they have a number of receptors and uptake systems for neurotransmitters. I've talked about the importance of potassium spatial buffering and the maintenance of the blood-brain barrier. And neuronal migration and proper end-target connection, which I actually skipped. It's very important to recognize, especially during development, that most of the cues or the signals that tell the neuron where it's destined to go originate from the astrocyte. The astrocyte elaborates various compounds that can diffuse and are taken up by the neurons, and the neuron actually migrates along the foot process of the astrocyte until it reaches its final destination. Ion channel cycling, contact between cells, removal of defective nerve terminals, removal of a number of noxious substances, and I'll talk about it a little bit later, immune responses, and metal sequestration are all functions that are important and exerted by astrocytes. In terms of this group, it's obviously interesting to mention metal sequestration. I don't know how many of you know, at least with respect to methylmercury, over time, about 80% of all the methylmercury in humans, as well as in non-human primates, accumulates within the different glial cells, and about 80% of it is found within the astrocytes. The same is true with lead. If you look at manganese, it's another compound that preferentially accumulates within astrocytes. So in terms of metal metabolism, metal detoxification, and effects of metals on these cells, there's clearly a great relationship that one needs to take into account. Now, the system that we use in our laboratory is a tissue culture. Most of the work that we do is tissue culture, and I just want to acquaint you a little bit in terms of the methodology that's associated in getting these cells. What we do is we usually take pup brains on neonatal day 1. We remove the brains out of the skull. We remove the meninges very carefully, and that's essential to do because the meninges contain fibroblasts, and fibroblasts will take over any kind of culture that you try to grow. They'll grow in a cup of tea. So you want to make sure that you remove the meninges, you dissociate the cells, and then you seed them in plates. You can do it either on glass cover slips or in petri dishes. And you can see that irrespective of the density of the cells or the seeding density, there are 3 different densities shown here, which are not very important, really, that initially there's a very large drop in the number of cells that remain viable. This is cells per plate. And after about 2 to 3 days, there's an exponential growth phase. The cells that attach to the bottom of the dish or to the glass start growing exponentially, and by about 2 weeks, there's a plateau. The reason for that is that the cells form contacts with one another, and that essentially tells them, stop dividing, stay happy, and that's the time that we use the cells. So we have a confluent, what we call a confluent monolayer. Now, how do we identify the cells that we have in culture? And again, I won't spend too much time on this. There are a number of proteins that are expressed within these cells, or a number of enzymes that are expressed within these cells. There are antibodies that are available commercially for these proteins, and one can just do immunohistochemistry or immunocytochemistry or some other techniques that allow you to visualize the cells that you have. Then you can go and count the number of cells that you have in the dish, and you can come up with the purity of the cells that you have. And obviously, if you're interested in astrocytic function, you wish to have high purity of cells. If you have 50% neurons and 50% astrocytes, you're in trouble, because when you look at the effects, you really don't know if the effects are exerted at the level of the neuron or at the level of the astrocyte. So culture purity is very important. And if one does this kind of staining, in this case we use glial fibrillary acidic protein, which is an intermediary cytoskeletal protein which is expressed in these cells and only in these cells. You can see very nicely, and in this case these are astrocytes from the retina, that you get beautiful cells, and actually if you were in focus it would look even better. You see a lot of cells that have the cell bodies with the various extensions that I've talked before. So this is a culture of astrocytes. I'll give you another example. In this case, we look at astrocytes and neurons. You can use dual labeling. You can label the neurons for proteins that are expressed specifically within the neuron, and then use another antibody for a protein that's expressed within the astrocyte. And in this case, we have an astrocyte here, and you can sort of see the relationship of the astrocyte with the neuron, and you have a number of neurons sitting here at the bottom of the dish. Okay, I want to switch now, after talking a little bit about the astrocytes, and give you a background about the end product that we were interested in. We were interested in the expression of a protein that's known as metallothionein within the astrocytes. This is taken from the internet. I can't give credit to whoever put it there because I don't know who did it, but it's not my slide. It's pretty though. The metallothioneins are proteins of molecular weight of 6 to 7 kilodaltons, and they contain about 60 amino acid residues. 20 of them, about 33% of the amino acids, are cysteines, and cysteines have very high content, or cysteine contain SH groups. They contain thiols, and as you all know, at least in terms of mercury, that becomes very important because mercury has a very high affinity for SH groups. The log association of mercury for SH groups is in the order of 10 to the power of 23. It's an extremely high number, and chemists or biochemists actually who are trying to isolate thiol groups or SH groups in a test tube commonly will use mercury to do the procedure. So obviously the potential here is that a compound such as metallothionein, a protein like metallothionein, may offer a buffering capacity for mercury. All of these cysteines are in their reduced form. I won't go into too many details, but suffices to say that the metals can bind to these cysteines, and usually there are 7 metal ions bind to one molecule of metallothionein. Again, it's a great buffering capacity for different metals that have a high affinity for SH groups. Now, what are the functions of the metallothioneins? Storage and regulation of cellular copper and zinc metabolism in response to dietary and physiological changes, control of fetal development, storage for the demands of cell differentiation and proliferation. Probably the most important function, especially during development. cellular adaptation to various types of stress, and I'll show you a slide in just a minute of various chemicals as well as physical conditions that induce the expression of metallothioneins in a number of cell types. The metallothioneins are important in metal transfer, and some recent studies suggest that metallothionein may be a very effective antioxidant. And again, that becomes important when we talk about mercury, because some of the theories about mercury toxicity are believed to be associated with production of oxygen radicals such as superoxide and hydroxyl radicals. So again, potentially the ability of metallothionein to scavenge free radicals bear some relationship to mercury toxicity. And finally, there's interest in this compound, especially the MT3 isoform, which I'll mention again in just a minute, because it's been recently shown that in patients that suffer from Alzheimer's disease, one cannot detect the expression of MT3. And that had suggested to Uchida's group in Japan that MT3 may function specifically in the regulation of neuronal survival. There are ongoing studies on this, and obviously it's a very hot area in neuroscience because it may afford a link between Alzheimer's and an etiological or a possibility that something dysfunctions in terms of the expression of MT3. Now, in terms of the distribution of the metallothioneins, the MT1 and the MT2 isoforms are the ones that are almost exclusively expressed within the astrocytes. There are some other studies that have looked at the expression of these two isoforms. You can find some in ependymal cells. There is some in arachnoid cells and in the pia mater. There's also a little bit in neurons, but most of the MT1 and MT2 is expressed in astrocytes. The MT3, the one that I just mentioned with respect to Alzheimer's disease, is exclusively found within neurons, especially neurons that have high zinc content. Now, if you look at this table, I think it becomes pretty apparent right away that metallothionein is just a stress protein. There are a number of physical conditions and a number of endogenous molecules as well as chemical compounds that have been shown to induce metallothioneins. The rate of induction is different amongst different tissues, but you can see that there's a wide array of chemicals and physical conditions that will induce expression of metallothioneins. When we speak about the metals, they're very potent inducers of MT1 and MT2. So they're inducers of astrocytic metallothioneins, but they're not good inducers, or they don't induce actually at all metallothionein 3, the one that's found within neurons. Just generally, a stress protein, just like heat shock protein, and all these conditions will affect the expression of metallothionein within a cell. Now, how does this happen in terms of, or how is metallothionein regulated in different cells upon exposure to metals? And I won't go through big details. I just want to show you that we are able now to understand those processes at the molecular level. And you all hear about the genome projects and a lot about molecular genetics and molecular biology. Many of these areas here that are shown as MREs, or metal regulatory elements, have been sequenced, and we know exactly what it takes to activate these areas and induce the metallothionein gene. This is known as the promoter region of the gene. This is the initiation box, and what I've shown here is the different metal regulatory elements. You can see there's about 17 base pairs, and there's a core of about 7 nucleotides here. It doesn't really matter what they are. And what happens is that when the concentration of the metal in the cell increases, it affects an MRBF, or a metal regulatory element binding factor, or a metal regulatory binding protein. This is like a sensing device that you have in the cell. When the concentration of the metal goes up, you get a conformational change. As you can see, I've switched the arrow from this way, going this way, to the other way. And when this sensing device changes the conformation, it binds avidly to the metal regulatory element. When they touch each other or get together, this is the signal that tells the gene here, okay, it's time to express yourself, start making some metallothionein. Now, I don't need to dwell very much about mercury, I think, in front of this group, but I just want to point out a couple things. First of all, the rate of oxidation of elemental mercury or mercury vapor is sufficiently slow. If you inhale it, not all of the mercury gets oxidized right away in the blood, and therefore, because it's a diffusible gas, some of it will get readily into the brain. There have been many studies looking at the uptake of inorganic mercury in the form of mercury chloride, for example, versus mercury vapor, which is also an inorganic form. And if you dose animals with equimolar or the same concentration, the amount of mercury in the brain after mercury vapor exposure is significantly greater than if you expose the animal to the mercury chloride species. The reason for that is that mercury chloride does not cross the blood-brain barrier very well, or not at all for that matter. And mercury vapor, before it gets oxidized, readily crosses into the CNS. So you get much higher concentrations. Once it's within the CNS, it's oxidized by way of catalase in the presence of hydrogen peroxide to form the inorganic mercury species, Hg2+, or mercuric mercury. That's the one that normally does not cross the blood-brain barrier. But once it's in the brain, it also is not very readily extruded from the brain, so it's essentially trapped in there. Okay? And what it does, because of the high affinity that it has for thiols, it readily binds to different SH groups in cells. And these are some of the symptoms. I think all of you are aware of those. There's no point for me really to reiterate those symptoms. I don't have to say very much about this either. I think you know about it. And you also know that there's a correlation between the number of amalgam-filled teeth and the concentration of mercury in the CNS, as well as in a number of exposure media, indicator media such as urine and the plasma. Now, the specific aim of the study that we got involved in was to look at the ability of astrocytes to sequester mercury. And as I said before, most of my work has been with methylmercury, but we got interested after I met Fritz Lorscheider at dinner in where was it? In Hot Springs? Yeah, in Hot Springs a couple years ago, or 4 years ago now, I guess. We started talking about our work, and we found that we had a lot in common. So I just asked him to send us some of these brains of the animals that have been exposed to mercury vapor, because we just don't have the apparatus to expose the animals. And we've been collaborating for the last few years. So the propensity of astrocytes to sequester mercury and their abundant expression of metallothionein 1 as well as metallothionein 2 led us to examine the ability of in utero mercury exposure to induce MT1 mRNA and MT2 mRNA. I'll show you some of that, as well as the proteins themselves in astrocytes. The second aim of the study was to assess whether induction of metallothioneins is a long-lasting response, thus potentially serving as a biomarker of mercury vapor exposure. In terms of the methods for this study specifically, Fritz exposed them for 4 hours a day from gestational day 7, this should be a 7, not a T, through gestational day 21. So it was a total of 14 days, and the level of mercury was 300 micrograms per cubic millimeter air for 4 hours a day. On day 21, Fritz removed the brains, put them on chilled gel packs, ice packs, shipped them Federal Express to us, and we got them within less than 24 hours. And what we did with those pups, or the brains of those pups, I should say, we did a number of studies using different techniques to look at the expression of the message the mRNA from metallothionein and the expression of the proteins themselves. And northern blots tells you, and I won't go into that technique, essentially how much of the message is there, how much mRNA is expressed in these cells. And obviously the presumption is that the more mRNA is in the cell, the more protein the cell is making, although that is not necessarily the case. We also did Western blot analysis, which essentially tells you how many proteins, or what's the level of protein that's expressed within the cell. And we did immunohistochemistry, which is just essentially using antibodies for the metallothionein and visualizing them with secondary fluorescent antibodies. Essentially, it's just complementary to the Western blots because, again, you're looking at the expression of metallothionein at the protein level. So again, both protein level and the message for the protein were looked at. This is a slide from Fritz. He has done the Mercury analysis on these brains, and what you see on the y-axis here is nanograms of mercury per gram tissue in the maternal brain, and on the right we have the fetal brain. Please note that this is a log scale, so there's actually very large differences between the controls, which are the open bars, and the mercury vapor exposed animals. It goes from about 20 to 30 nanograms of mercury per gram in the brains of the controls to about 800 to 900 nanograms mercury in the brains of the dams that were exposed to mercury. And you can see that there's essentially the same kind of relationship in the fetal brains. The levels in the controls were a little bit lower, about 8 nanograms of mercury per gram, and in the treated pups all the way up to about 100 nanograms of mercury per gram tissue. Now, as I described earlier, what we did with these brains was to isolate the astrocytes, grow them in the dish, and then we did the northerns and the westerns to look at the message and the protein. Essentially, we just isolated the cells, we seeded them into the plates, and we did not touch them for the next 3 weeks. We had a control group and a mercury vapor treated group. So they were just maintaining culture under the same conditions in the same incubator. And at 1 week, at 2 weeks, and at 3 weeks, we have looked at the expression of the message. We looked at the message mRNA for metallothionein, both MT1 and MT2. And this would be a characteristic northern blot. And let me just tell you what the different lanes are. Lane 1 is control at 1 week. Lane 2 is mercury vapor at 1 week. Control at 2 weeks. Mercury vapor at 2 weeks. Control at 3 weeks. And mercury vapor at 3 weeks. And I think that you can, just by looking at visually, I think you can appreciate that in the mercury vapor treated pups, or the astrocytes that were derived from the mercury vapor treated pups, there's a higher density. The band is a lot darker, which essentially means that there's more mRNA expressed in the mercury vapor treated astrocytes. And what you can do with these kinds of blots is scan them and then do some very sophisticated densitometry and analyze the data and express them quantitatively. And what I've done here is simply standardize the expression of the metallothionein mRNA to control levels. So what we have here is empty mRNA abundance, percent relative change, and the controls at 1 week, 2 weeks, and 3 weeks are shown as 100%, and then MT1 at 1 week in mercury vapor, in astrocytes derived from mercury vapor treated pups is here, 2 weeks here, and 3 weeks here. And you can see that statistically, all of these are significantly different from their respective controls. There were no differences in terms of MT2 mRNA expression at 1 and 2 weeks in culture, but there was a significant increase at 3 weeks. And the reason for the fact that we don't see any change here is simply related probably to the fact that the message for MT2 mRNA is a lot lower compared to MT1 mRNA. We just can't pick it up very well. But clearly, you can see that just taking these cells out of the brain, seating them into a dish, not touching them for 1, 2, or 3 weeks, even though they have not been treated for 1, 2, or 3 weeks after they were removed, we still find increased levels of expression of mRNA. This is essentially the same thing, but it was done on whole brains. This is another mRNA blot for MT1. It was very hard to get the message because, as I said before, most of the MT1 and MT2 is expressed only in astrocytes. And when you homogenize the whole brain, you get a lot of other cells and you get a lot of noise. There's a lot of other cells that don't express the message. So the astrocytes are only a small part of the cells that you isolate. And therefore, because of the noise, it's very hard to get a very nice and crisp message. But I think you can still appreciate visually that in the brains of the mercury vapor treated animals, there's more message. And if you look at it quantitatively, again, we have empty mRNA abundance, 100% in controls. There's about a 50% increase in the message in whole brains of pups, and these would be gestational day 21 after 14 days of mercury vapor exposure. This is a Western blot, so what we're doing here is we're looking at the amount of proteins in these cells. And what we have here is just some standards that one can buy commercially from a company such as Sigma, for example. We isolate the proteins, we then run them on a gel, SDS gel. The proteins migrate down, and you can run the standards with them, and that allows you to verify that actually what you have is actually the protein that you're interested in when you stain them. And here we have controls and mercury vapor derived astrocytes at 1 week in culture, 2 weeks in culture, and 3 weeks in culture. And again, I think you can appreciate that at least in 2 and at 3 weeks, the amount of protein expressed in the astrocytes derived from the mercury vapor treated astrocytes is greater than their respective controls at 2 and 3 weeks. So there's a good correlation between what we find at the level of the mRNA and the expression of the protein itself. This is the immunohistochemistry, which just essentially corroborates the protein data that I've just shown you on the Western blot. What we have here is astrocyte cultures that have been stained with antibody specific for MT1 and MT2. And then the message is amplified by using a secondary antibody that's conjugated to fluorescein. And we can visualize the fluorescein under the microscope, and it looks green. So these are control cells, 1 week in culture, derived, again, from control, astrocytes. These are the mercury vapor at 1 week. And these are the cells at 3 weeks. Very little metallothionein expression in these cells, although you could see it. And a marked increase in expression in cells at 3 weeks in culture that are derived from mercury vapor treated animals. Again, corroborating what I've just pointed out in terms of the Westerns in the previous slide. So in summary, we can say that in utero mercury treatment or mercury vapor treatment leads to an increase in metallothionein 1 mRNA in whole brains, as well as metallothionein 1 and 2 mRNA and proteins in astrocyte cultures. These studies provide evidence that mercury vapor is a potent inducer of metallothionein 1 and MT2 gene products in developing brains. Furthermore, exposure to mercury vapor has long-lasting effects. As I said, in the astrocytes that we've isolated and we had grown in cultures, the levels of metallothionein are persistently higher compared to those in matched controls for up to 3 weeks in culture. And we really can't go far beyond that because the cells just don't survive. But at least until 3 weeks in culture, there's definitely a big difference between the controls and the mercury vapor treated cells. It would appear, therefore, that the metallothioneins are useful quantitative biochemical marker of mercury exposure. Now, I want to finish up with some recent data that we actually got over the last probably couple months that deal with an enzyme that's known as glutamine synthetase, which is shown here. this enzyme is again astrocyte specific. It's an interesting enzyme because it requires manganese. I don't know if you can see it. Manganese is here in sort of light blue. It's a cofactor. And as I said before, about 80% of all the manganese in the brain is found within the astrocytes, which are the only cells in the brain that express this. The function of this enzyme is shown in this schematic. Just for orientation, what we have here is a presynaptic terminal of a neuron and a postsynaptic terminal, the same here. And it just shows you the cycle of glutamate, glutamine, and gamma aminobutyric acid within the central nervous system. These are all essential neurotransmitters. Glutamate is packaged in vesicles. In the neuron, it's released into the extracellular space, and then it's taken up by the astrocyte. It combines with ammonia, and this reaction is catalyzed by the enzyme that I just mentioned, glutamine synthetase, to form glutamine. This is a noxious substance, actually both of these. Glutamate will excite receptors on neurons. You may have heard about N-methyl D-aspartate receptors or NMDA receptors. If there's too much glutamate in the extracellular fluid, it will excite the neurons and eventually they die. So it's imperative that the glutamate be removed and then recycled to glutamine, and this enzyme again, glutamine synthetase, has catalyzes this reaction. Subsequently, glutamine can go back into the neuron. It's taken up by the neuron. It can form back glutamate and GABA aminobutyric acid, which is an inhibitory amino acid, and that can be then recycled again within the astrocyte. So you can imagine that if something happened to this enzyme here, you would obviously affect the neurotransmission as it relates to a number of these neurotransmitters, glutamate, glutamine, and GABA. Now, what we've done over the last few months is look at the expression of glutamine synthetase, mRNA, in the same brains that we got from Fritz Leuschheider. The techniques are essentially the same, at least for this slide. What we have here is a northern blot where we are looking at the message of mRNA in the brains, and these are whole brains of animals. So these would be gestational day 21 rats that have been exposed to mercury vapor. And I think you can appreciate the glutamine synthetase is right here, these two lanes. These are two controls, and these are two animals that had been treated, and you can see a marked increase in the amount of the expression of glutamine synthetase. We've also done the same thing in the tissue culture, and we're using a relatively new technique, which I won't go again into great detail. You may read about it in the future, so remember me when you read about it. It's reverse transcriptase RT-PCR. PCR stands for polymerase chain reaction. And essentially what is being done is you isolate the RNA from the cell, and then in our case we constructed specific primers, gene-specific primers that form a template that's complementary to the RNA, okay, and it forms a cDNA. This is the way we can synthesize the cDNA. The polymerase chain reaction then consists of a thermorecycling. You just do it repetitively for about 30 or 35 cycles, and what it does, it just amplifies the message. And the advantage is that you can take a very small, or if you have a very small amount of RNA, you can amplify the message so that when you run it on the gel, you can see something on the gel itself. If you just do a northern blot, sometimes you may not see that, and we've had problems just by running northern blots because we just didn't have enough mRNA from the tissue cultures. So when we do that, and I know it looks scary, this is essentially what you get. I'll just focus on these 4 lanes here. What we had done is we used 2 different gene primers, one for glutamine synthetase, which is the enzyme that I've been talking about, and one for beta-actin, which we use as what's known as a housekeeping gene. it allows us to standardize the levels of glutamine synthetase that we get to an enzyme or a protein that's found within the cell that one would not suspect would change with the mercury vapor treatment. And therefore we can standardize the amount of GS to the level of beta-actin that we find within the cell. Now, excuse me, what we have here are 4 lanes, and these are again, this is the RT-PCR product that was isolated from tissue cultures at 2 weeks, these 2 lanes, and at 3 weeks. And these are the controls, and these are the mercury vapor. This is the GS, and this is the beta-actin over here. And I think, at least for the 2-week cultures, I think you can appreciate that, especially if you standardize the beta-actin levels, if you, for example, double this to match what you have here, you get a tremendous increase in the amount of GS within these cells. And there's somewhat of a difference here. I know it's hard to see. It's certainly not as striking as the difference that we see at 2 weeks. So it may actually come back to normal at 3 weeks, but definitely at 2 weeks there's a clear change. Now these are preliminary results. We'll definitely have to do it a few more times, but it suggests to us that the mercury vapor may also have an effect on glutamate transmission, potentially. Now I want to finish by thanking some of the people that I've been working with. Kelly Cohen and Dawn Conklin. Kelly is gone actually, she left for Pennsylvania, but she was a technician in my lab. Chengping Yao is a postdoc in my lab. Ken Vrana has helped us with some of the molecular biology. As I mentioned, Fritz had kindly supplied us with the animals. And I also want to acknowledge Dr. Tan, which is not on the list here. He's a collaborator from Winston-Salem State University that helped us with the RT-PCR. And you may have seen the last slide, but I couldn't resist it. It won't go. Yes, it will. What do you mean you're not going to school? What's your excuse? My tooth fillings contain mercury. I'll stop here.
Before the break, those who are going to take the exam, please see me at the door. Thank you.
Our next speaker really doesn't need an introduction to our academy, but once you've heard his presentation, I think you'll agree how beautifully his presentation will dovetail with what you've just heard. The microanalytical techniques that are used here have been Boyd is going to share with you some of those techniques and methods in the next presentation. Am I correct in saying that you are now chairman of the Department of Chemistry at University of Kentucky? Is that correct? Boyd, where are you? Is that correct? Good. That's happened, I believe, last year. Let me welcome from the University of Kentucky, Dr. Boyd Haley. You're going to enjoy this.
Start talking as soon as I get everything working. What you'll find is that Dr. Aschner's work is very exciting to me because it dovetails more than just a little bit into what we've been doing. Several years ago we reported in the Proceedings of the National Academy of Sciences that you could probably diagnose Alzheimer's disease by elevated glutamine synthetase released into the cerebral spinal fluid. And other things he's done is right down the same alley of the work. We're more biochemical and he's more cell biology, but it was really very useful to listen to his talk, and I was quite excited about his results. And it again is another demonstration of how mercury mimics the effects that we see in Alzheimer's disease. What he's showing in Alzheimer's disease is what we see, and what he's showing in mercury-treated rats is what we've seen in Alzheimer's disease. Okay. So what? Oh, okay. me now? Can you hear me now? Good. There's no way that I can teach everyone in this room the peculiarities and the fine points of photoaffinity labeling. It's a high technology. It's something that I started doing when I was a graduate student and a postdoc about 24, 25 years ago. And so throughout this thing, I'm not going to try and belabor the chemistry. It's even difficult for biochemists who have not had chemistry for a while to understand. But I'm going to tell you what I'm going to tell you, and then we'll go through the whole process. Using this technology, we can look at proteins that are missing or proteins that are present in diseased tissues versus normal tissues. We can also use this to detect changes in important, metabolically important enzymes that are exposed to toxins such as mercury. And the first example, so you can get with me right from the very start, then what you have to remember is if you have a control, and you see this protein right here, if we treat this protein with mercury, increasing concentrations, and we're talking about concentrations at 10 to the minus 7th, 10 to the minus 6 molar, we can abolish this protein and very selectively and have very little effect on the protein underneath. The two here to the right, this is a control, and you see we wipe this protein out with increasing mercury. The same thing with this control, 0.8 times 10, 8 times 10 to the minus 7th molar mercury, takes that down about 90%. Abolishes it if we go a little bit higher. And if you look, you can see that over here with our two Alzheimer's disease controls, we've made normal brain just by the addition of a little bit of mercury look just like an Alzheimer's disease brain. So this is what we're going to talk about, is how we can do these studies and can we look at mercury toxicity and see how it affects the different cell systems. And we want to talk about how important this is to the body. The thing for some of you who are sitting here who don't understand photoaffinity labeling, and you never will, and it's not something you have a chat about and understand, I would point out that we've been using this. This paper was done in 1977 in my lab when I was at the University of Wyoming as in the National Academy of Sciences, and it talked about the properties of how you could use this compound, the photoaffinity probe, to study tubulin biochemistry. We showed that it could substitute. It's a mimic of the natural compound. Later on, we used it to localize the binding site on the specific beta subunit. 1983, we demonstrated that you could use this. The hydrolysis of this occurred when tubulin was polymerizing. And in 1994, with the postdoc in my laboratory, we identified the GTP binding site on the protein and showed that it contained a sulfhydryl, which is probably the reason why mercury inhibits its binding. The whole list of this is just to show you that this is credible research. This isn't something that just came up the day before I first met you folks to start talking about mercury toxicity. It's accepted and is published. These are all very good journals. The Journal of Biological Chemistry and the Proceedings of the National Academy of Science and Methods in Enzymology are about as good a journals as you can publish biochemistry research in. The technology is really simple once you make the compound. The compound and the synthesis of the compound is quite difficult. And if you don't believe me, ask Dr. Pendergrass and Dr. Ashok Chavan, who worked for me. This is something that's very intense, and you don't take coffee breaks when you're doing this. But once you make the compound, you can take a brain homogenate or any tissue homogenate, including curvicular fluid from near an infected tooth or a control tooth. You add the radioactive compound in here. This will bind to specific proteins. And I would point out at this time, we have several different analogs, not only of GTP, but of ATP, cyclic A, cyclic G, etc., that we can test. If you incubate this for 20 seconds at 4 degrees, allow this to bind to specific proteins, shine light on it. It's kind of a Star Wars type of biochemistry. The compound literally explodes, giving off nitrogen gas, and this radioactive compound will selectively covalently link to any protein that binds it. And so we see normal proteins bind it, and if it's healthy, we see a big black band. If we kill all the protein, we get no labeling because it doesn't bind once you denature a protein. So this is the technology. We'll talk a little bit about tubulin. Tubulin is the major protein in brain tissue. It is the major protein found in neuronal cells. It is the railroad track of the cell. And tubulin, can somebody focus? Tubulin starts out as an alpha-beta heterodimer, a small protein with its building blocks, and it requires the binding of GTP to form this polymer, or this microtubule, or microfilament, that looks something like this. And if you were to look down from the top, the direction that arrow is, you would see it's a hollow core and it's cylindrical, and it goes from the nucleus to the plasma membrane. And in a neuronal cell, it goes all the way down the axon, and it is the railroad tracks on which you carry things like the neurotransmitter glutamate. So this is the biochemistry. And if we look at this in a cartoon that you use when you're teaching graduate students or some neurophysiology, if this were a neuron, this would be the nucleus where a lot of synthesis takes place. Anything that's made here has to get to the end of one of these axons. And so if we go to the cutaway here, it has to rotate and be attached, packaged, collected onto a motor, and migrate down this axon. If we blow this section up, you can see this is tubulin. And on here we set little molecular motors that bind another nucleotide. They bind an organelle such as this, which could contain glutamate, for example, or certain other things, and it actually migrates down this railroad track, traveling from the nuclear region down to the synapse for release. There's another molecular motor called kinesin. Some of them run this direction, and others run the opposite direction. We call it retrograde and anterograde transport. So tubulin is quite important. Now, if we try to look at how it appears in the cell, if we do an immunostain, you can see that all of this yellow through here is microtubules that are running through a neuronal cell. So it's in very high concentration in all cells, not just neuronal cells, but in all cells. So this is your railroad tracks. Now, if you're thinking that you need all of this, or the cell wouldn't make it, although there is quite a bit of redundancy, how would you like to have 10% of the roads torn up going out of your city, or make it 50%? Or 60%. You don't have to wipe it all out, and what we're talking about is how much mercury it takes to take it all out. You don't have to take it all out to impair a cell. You just have to take out enough to disrupt its function. But you can see that it's at quite high concentrations, and this is a protein that you would not want to mess up with or have problems with if you're trying to keep your brain or your nerve cells working properly. The kind of screen we do in the laboratory, and the people here, Dr. Ashok Chavan and Dr. Kurt Pendergrass, have done tons of these studies, and Kurt's done most of the mercury studies that we'll talk about here. If you look at my papers, I always put my name on last. Kurt's name is almost always first because he did the work, and he understands this as well as I do. So the material I'm talking about, he deserves every bit as much credit as myself. But the kind of screen we do, this is just a gel. After we do these experiments, we solubilize the protein with a material called sodium dodecyl sulfate. It's the active ingredient in Tide. So it just takes the proteins and solubilizes them. We put them in electric current, run them down through a gel, and they separate on molecular weight. As you can see, here are our molecular weight standards on the outside. When they're in the gel, you can put the gel in with a dye that stains protein, and every protein band in here will be turned purple or blue, depending on how your vision works. And as you can see, we've never been able to find any major changes between Alzheimer's disease and human control disease brain protein profiles from the massive proteins, although you'll find every now and then one that's unusual. It doesn't hold up. And our controls, by the way, aren't, they're people that have died of other reasons and sometimes other neurological diseases. If we now expose this after we photolabeled, we can expose this to a gel, or pardon me, to an X-ray film, and we can tell whether or not the proteins, there's been a change. And routinely, this is just one of the samples of the things we've done, and always in control and every other neurological disease we've tested, we find that beta-tubulin is there at a significant level, and we can photolabel it. However, if we go to Alzheimer's disease going across, you can see in most of them it's dramatically decreased. In certain ones, we see they're almost at control levels, although they're quite a bit dropped, and many times we find that Alzheimer's disease is a tautology. You diagnose it by progressive dementia and the appearance of neurofibrillary tangles and plaques. There's also another disease called diffuse Lewy body disease, and this is the one that's sometimes confused with Alzheimer's disease, and many times we will go to the Alzheimer's Center when we do this before we get the pathology report and talk to them, and invariably we'll find out that some of these are not Alzheimer's disease. They'll call them Alzheimer's/diffuse Lewy body disease, because there is no known cause, no known etiology of Alzheimer's disease. It's a clinical observation along with a pathological analysis of the brain that is a tautology, because a lot of brains, a lot of people will die with a tremendous amount of senile plaques, and they don't have Alzheimer's disease. They had no clinical dementia. So it is something that is still questionable. And this is just a slide I got from Kurt talking about the diagnosis of Alzheimer's disease. You have this clinical diagnosis, which is just a battery of exams. You can have MRI and PET scans, which are quite expensive. And basically the way you say it's Alzheimer's is you rule out all other possible causes of dementia. And they still can't say you have Alzheimer's disease until you die. They remove your brain and they do the pathology and they see the senile plaques and the neurofibrillary tangles in excess of normal aging. Because a lot of people have these. We had one person that was, I think, 98 years old. He was not demented. He was a professor that graduated from the University of Kentucky, and his hobby was collecting PhD degrees. And I think he had picked up several degrees, 3 to 7, I can't remember the exact number. When he died, his brain was loaded with senile plaques. His tubulin photolabeled, and he was clinically normal. He was not demented at all. So you can't use some of these as the effect. Now, when we saw this problem that tubulin does not photolabel, tubulin is totally dysfunctional in Alzheimer's disease. And thinking logically as a scientist, the most sensitive protein that I know of that can be affected by toxins is tubulin. It's very easy to have tubulin become abnormal or abnormally precipitate or aggregate in a test tube. So we set on a search to find out what could possibly mimic this, what could be an additive factor, or what could be the toxic material that could cause or aid or exacerbate the condition that we could describe as Alzheimer's. So we started looking at heavy metals. So we're going to talk first about the effect of heavy metals on brain nucleotide binding proteins, and then we're going to talk about the same effect in cerebral spinal fluid nucleotide binding proteins, because this does lead to a diagnostic test that we can possibly use. We did a screen. We did several screens. Kurt will tell you, I make people do things over and over because I don't like being wrong, especially if I'm going to publish it. And we did this with several metals, with and without EDTA. And what we found is that mercury added from the outside in the presence of EDTA was the one that gave the most potent inhibition of tubulin function. Most metal ions would do nothing, absolutely nothing to the photolabeling. And we published this. It's been published several times, and we've followed this up. And the more dramatic effect is shown here. And this is an experiment where we're looking at beta tubulin in a controlled brain, and we're adding mercury. We have EDTA in all of these samples. We do it, half of it without magnesium. This is done for a reason that's too complex to explain here, but magnesium is involved in a binding site on tubulin. And then we put in 3 micromolar mercury. We let it set for 5 and 60 minutes on our bench top, and you can see there's no change in the photolabeling. If we added the mercury, we waited 2 minutes. It did very little at this time. It dropped it a bit. But if we waited 5 minutes, it totally wiped it out. So the inhibition is time dependent. The more the mercury sets there with it, the more it will inhibit. We did the same thing over here by having magnesium present, and you can see it slowed down the rate because it took, out here 3, we went out here to 35 minutes, but at 35 minutes, it got it all. So you can slow down the rate of mercury, but if it's in your brain, it's going to settle in on tubulin if it's not bound up by something very specific. And I think that over here we have without mercury, again for 5 and 60 minutes, plus EDTA, and you can see that this still photolabeled quite effectively. So there was no doubt, and we've published a lot on this, so I'm not going to beat it to death too much, but I do think we take the take-home lesson. There are a lot of people in this country that are on that thin red line between being normal and going over that step where you go into dementia, Alzheimer's disease, whatever dementia, whatever form you want to cause it. So someone can argue and say, mercury does not cause Alzheimer's disease. I have not proven that mercury causes Alzheimer's disease. We don't make that claim. But anyone that would look at this data and would say that someone who's susceptible to Alzheimer's disease or near that thin red line would best be served if they didn't have mercury in their mouth or mercury going into their body under any condition. That's a no-brainer. That's a simple thing to say, and I have no trouble selling this to the best medical schools. I probably would have trouble selling this to certain dental organizations. I don't think I have trouble selling it here. And this is the bottom line here. And again, it's the one you saw at first. Again, 2 ADs, 2 controls, and if you increase the mercury levels and you don't have to go very high, you can make a control have the same biochemical properties as an AD. And Dr. Pendergrass did this work, by the way. And when I gave this talk, when I first was drug into this organization by Mike Ziff, who found my paper somewhere, and I don't know how I located it, but you all remember my first presentation. It was a lesson in miscommunication, I think, because I thought you were all biochemists or toxicologists. but I was sitting, the second talk I gave was in Germany, and I was sitting there listening to this fight. It was is amalgam good or bad, a discussion that was held by the European Academy of Science, and Murray Vimy was there. And in the process of looking at it, I realized that these people, many of them were looking at mercury like all mercury's the same mercury. Mercury vapor is a totally different species. It behaves totally different, and you cannot say so many micrograms of mercury is toxic. What kind of mercury are we talking about? And so with this discussion, I made an agreement with Murray, and we started the next experiment, because the real criticism of my work, which I would give, is that you're doing it in a test tube, in a tissue homogenate. And can you say that the mercury gets from the vapor in there? And of course I knew that, but in science you do a lot of experiments to convince the idiots. With everything I read, anybody would say mercury gets in the brain. That's a no-brainer. But we had to do the experiments, show it would get into the brain and affect tubulin. We couldn't find anybody to volunteer for the experiment, so we had to use rats. But the basic aspect of this thing is that if you have mercury and you're breathing it, it gets into it. Some of it can be taken into the bloodstream, converted to the +2 cation, and what it does is it either tears up your red cells or goes into your kidney and causes problems, or liver. But however, some of it has to go right directly through the nasal mucosa and go directly into the brain before it gets converted to the Hg2+, and that's the reason mercury builds up in the brain tissue. And in this aspect, it will react with the sulfhydryls that are on specific brain proteins such as tubulin. And this experiment was done with Murray Vimy and Fritz Lorscheider. The hard lab work was done by Dr. Pendergrass. But the first part here, the exposure of the rats to mercury vapor, was done in Calgary, and the analysis of mercury in the brain was done, I think, in Calgary. The rest of this, separating the brain proteins, the autoradiography and the photolabeling, and the densomagnetic scans of the autoradiograms to quantify the labeled protein, was done at the University of Kentucky. This is the chamber that they built. I didn't build this. Fritz Lorscheider has this. He sets a rat in here with the fluorometers, with the flow meters, and the exhaust being pulled out by the vacuum pump and charcoal traps. He can, pump and suck mercury vapor off of a mercury vessel into this thing at a measured rate and determine what it is, so you can really carefully tell how much mercury vapor the rat's been exposed to. And this is the result. Again, if we focus in on this side over here, we see here's an Alzheimer's disease brain. It doesn't photolabel beta tubulin. Here's a tubulin brain. I don't know if Can somebody focus? I don't see a focus on this. Okay. At any rate, you can see our two control rats, the tubulin photolabeled very nicely. When we exposed them to mercury, and these were exposed to mercury, I think, for 2 weeks for 4 hours a day at 300 micrograms per meter cubed, which is it's high, but it's not any higher than what some people might have in their mouth if they were exposed. And what we found by doing this, Kurt can probably correct me, but I think we did this on 21 rats. And the bottom line was we saw anywhere from a 41 to 75% decrease in rats that were exposed to mercury vapor in the photolabeling of their tubulin. Now this protein up here is something different. Humans don't have it, so we can, if you send us rat brain versus human brain, we can tell the two apart. But the beta tubulin is definitely decreased, and it's a little bit different in each one of them, but all of them are less than what you would see in the controls. So what this said was that if we, if we take the data and put it out in bar graphs, because we can count this radioactivity. In all these bands, we have an instrument. It's called an Amaz Detector. If you put your gel in there, it'll tell you the radioactivity. And anything that you take a mouse from I shouldn't use mouse at this time. If you take a computer and draw a circle around it using your mouse, it'll tell you the amount of radioactivity that's in your circle or anything, so we can count it. And if we look at the control, they had very little mercury in their brains, but at 2 weeks, and their tubulin photolabeled very nicely. If we did a 2-day dose, we see that the mercury starts going up, but the tubulin is still photolabeling at the same level. This is probably because, as you heard Dr. Aschner talk, your metallothionein is working quite well. It's probably sucking it out and preventing it from getting to the brain or to the tubulin. However, if you go to 14 days, the mercury level gets up around 400 nanograms per gram wet weight at this level, and the tubulin photolabeling drops down to this level. making this look something like here's our control brain and here's the amount we saw in our Alzheimer's disease brain. So there's no doubt as the mercury level goes up, you see a dramatic drop when you reach a certain point in the ability to photolabel tubulin. And that's telling you that mercury is disrupting tubulin polymerization. It's disrupting tubulin, and that tubulin, of course, is what's disrupted in binding. Now, I want to go back. This, again, is some of Kurt's work. If you look at the level of mercury, 2 weeks, 4 hours a day, they put in over 400 nanograms per gram wet weight. That's a lot. That's just mercury vapor. Now, before we did this, we tried feeding rats mercury. And this is, tells you how much patience Dr. Pendergrass has. We were feeding them 70 micromolar mercury chloride in their drinking water. That's all they had to drink. We had control rats, and we had the mercury-fed rats, and they had nothing but this to drink for 12 months. And we looked at the amount of mercury that we get in their brain. And in 3 months, it jumped up this high. The controls were slowly going up, indicating that they were probably getting secondary breath from the rats that were getting mercury. But you can see it goes up here, and then after a period of time, it kind of levels off from 9 months to 12 months. And if you go across, and look, we only got to about 150 to 160 nanograms per gram wet weight. What that's telling you, and if you know, when we evolved on Earth, you know, when our knuckles dragging the ground, we ate bad food and we drank bad water. But we didn't breathe mercury vapor. We have protection here. All mammals do, probably. We can handle that. But mercury vapor is something that we have no protection against. We did not evolve much protection for that at all. And the difference is 400 nanograms per gram wet weight in 2 weeks versus 150 to 160 in 12 months by drinking the material. So it does indicate that the vapor, is, you know, as far as the brain goes, is much more disastrous. Again, I think it's really easy to attack somebody that's on a point by himself, and I think Kurt felt nervous out there sometimes. I know I did. If you think our data is out of the ordinary or something spectacular or something that you wouldn't expect, that's only if you're not really well educated in this area. This is just nothing more than a list of publications where people have looked at tubulin, not like we have, and maybe not in brain, but looked at the fact, for example, we just take one, I'm talking about 25 micromolar mercury, you know, gave, and mercury, methyl mercury, and then it took 250 micromolar copper and 500 micromolar cadmium, mercury by far the most potent, you know, caused the depolymerization of tubulin. And the IC50 was 3.8 micromolar and 7.6 micromolar for these two species of mercury. So there's really nothing very surprising about what we found. And I would be very honest with you, in this debate about mercury being toxic and getting into the brain and causing problems, I feel like I've been in a 4-year argument with the town drunk. This is really, and you guys should feel that way too. When the dentist and the ADA or the dentist, anyone that tells you mercury's not toxic and the amount that comes out, it can't be bad for you, just laugh at them. I mean, you know, you have to understand that it's really, the argument is that silly to any scientist. And the other point we want to talk about, you know, is that it's not tubulin. Tubulin isn't the only thing that's affected. It's the one that we've studied. We haven't studied an enzyme, and I don't think anyone has, that mercury doesn't affect. And if we look at this slide, again, this was prepared by Dr. Pendergrass, and I'd point out the top one, glutamine synthetase, is inhibited. It has an IC50 of 5 micromolar for mercury, so it's very much affected. It has a nice ATP binding site, and we have published on this enzyme and the effect of metal ions on the binding. Creatine kinase is one of them that you see in our batch of enzymes that we use as test enzymes, and it's 4.5 micromolar. And then let's drop down here and take a look at the sodium potassium ATPase, which is probably more important than all these enzymes to a living cell. If you talk about it, it's inhibited at a 0.2 micromolar. So while we're telling you that you lost all your creatine kinase in one of our assays, you can damn well bet that your sodium potassium ATPase isn't doing so well either. Since it's about 4.5 divided by 0.2, that much more sensitive to mercury as far as it being inhibited. So while we see just the tip of the iceberg with some of these things, with tubulin inhibition, et cetera, mercury's non-selective. It's which enzyme it runs into first. It has a sulfhydryl. That's the one that's going to kill. It's not a good hitman. It just kills everything in its sight. It's and these are just publications, and I would just point out this is work done by other people. Some of us, Kurt's work here with the AFGF, But most of this has come from other laboratories, so we're not alone in reporting mercury toxicity to certain things. Now, we want to go back and say, why would mercury be particularly effective in Alzheimer's disease? And there is this gene which people have identified. Now, I just read recently in the paper that this applies mainly to white people. So if you're white in the United States, this applies to you, and evidently it doesn't apply to minorities, that there's something else going on. But the APOE gene is very significant, and it's the one that I think Athena Neurosciences uses in its bank of proteins and identification markers to tell you whether or not you're susceptible or likely to have Alzheimer's disease. And in this gene, again, this is a slide made by Kurt, it's a genotype, APOE, and there are 3 forms. There's 2, 3, and 4. And the percent in the U.S. population is given in this line, and you can look at this. You can tell that most of us have 3. 2 copies of 3. We get 1 copy from our mother, 1 copy from our father. However, if we talk about the average age of AD onset that's been calculated by adapted from Rosen's 1995 Scientific American and Science and Medicine, if we look at this and we look at the ratio, what it comes down to is having 2 copies of 2, you're not going to get Alzheimer's disease. You're going to die of shriveling up and blowing away before it happens, more or less. But if you get down here 2 copies of 4 or a copy of 3 and 4, you're in the high risk range. 2 copies of 4, you're going to get it before you're 70 years old. It's almost a given, not a given, but I mean you're definitely at risk. 2 and 3, you'll be above 90. 2 and 4, it's a little bit lower. But the general consensus in the area today is that 4 is the worst to have, 2 is the best as far as protection against Alzheimer's disease, and 3 is intermediate. And if we look at the structure, when I saw this data and read this, I immediately went to the literature and we looked up the structure of the APOE proteins. And APOE2, which protects you, and you have to understand it's a housekeeping protein. It's made in your brain, released into the cerebral spinal fluid, goes into the plasma and is excreted from the body, taking out waste products that you don't want in the brain. That's generally what it's about. So this is its second highest concentration in the body is in the cerebral spinal fluid. And if we look at this, APOE2, which protects you, has 2 cysteines, and the only change between it and APOE3, one of the cysteines is converted to an arginine. This one has 2 binding sites to carry out mercury. This one has 1, and the APOE, which doesn't protect you much at all, has 2. Now, it doesn't mean that this is the major function of this, because it's not. It's a lipid-carrying protein. But it does indicate that if you're thinking and you're logically trying to put together a lot of circumstantial evidence of what might be contributing to Alzheimer's disease, you have to look at this. And other people, we propose that it's probably protective against things like mercury and cadmium and perhaps excess zinc. Other people say, no, this has better reducing power, but make a better free radical scavenger than with this, and they're right. So both of those could be taken into place. But they still can't argue. If you have cysteines floating around on the exterior of a protein, and for mercury to get to your brain, it has to come in contact with your CSF. This should give you some help. I'd like for them to prove me that it doesn't. Let's put it that way. So what I'm saying is that there's a lot of correlations, a lot of factors that say that mercury would put at risk anyone that has the genetic makeup or the exposure factors to Alzheimer's disease. You can't say it causes it, and I don't want any of you saying it. I mean, it may be. I mean, if you take enough mercury, you become demented. There's no doubt about that. But we can't say it's causal yet. But they can't argue with you with the fact that it would exacerbate the condition. And if they say, well, that most people can handle that, well, I don't care. What about those people that are near that thin red line? They would like to make it to 70 before they're demented instead of going there at 65. And I don't see how anybody can argue against that. So we're now going to switch gears here a little bit. We started looking at cerebral spinal fluid, and we have seen differences that are very consistent in the cerebral spinal fluid of Alzheimer's disease patients versus non-Alzheimer's disease patients. The difference is people who have AD do not get lumbar punctures. They do not get lumbar cerebral spinal fluid drawn from the lower back. All of the samples we got, or most of them that we obtained, were from the ventricular region, which is after they die, when they open up the head to do the autopsy on the brain, they would stick a needle in the ventricular region, which is in your brain, buried in your brain, and pull out those samples. And we saw differences in the nucleotide binding proteins of that fluid. And the major difference we saw at one time under one set of conditions was the fact that there was elevated glutamine synthetase in the brains of cerebrospinal fluid of AD patients, which Dr. Aschner just showed you is elevated by exposure to mercury. So they may both be responding to the same response, that is, tubulin doesn't polymerize, but it's definitely there. So we started working on this, and we got frustrated because we couldn't get lumbar CSF from AD patients, so we started working on Lou Gehrig's disease, or amyotrophic lateral sclerosis. And what this is, we have another technique. We can take proteins and separate them by their molecular weight. But if before we do that, we put them in a current and we separate them based on their charge, we can separate them this way based upon the total charge they carry, whether they're a basic protein or an acidic protein. We can lay this whole gel on top of here and come down this direction and identify proteins because they'll migrate down this way with molecular weight, and their position in this direction will be based on their isoelectric point. Now, if you look at this below 30,000, and this is what most people have used when they've tried to diagnose certain neurological diseases, there's not many proteins that you can see below 45,000 even. You can find a few of them, but up here you see a lot of little dots that represent specific proteins that are normally seen in cerebral spinal fluid. And this is a stained two-dimensional gel of an ALS CSF. We couldn't see any changes in the proteins, none at all. However, if we photo-labeled this and we ran this around, ran this gel, you could see that on controlled cerebral spinal fluid, photo-labeled with the ATP, that we always saw this band here. And you can look at this spot, you know, where the arrow's pointing, and you can see the constellation that surrounds it, plus this strip that goes across the top of low 31, which you couldn't detect. And if we refine this, you would see that this is all several, probably 30 proteins that we've photo-labeled, that if we didn't expose this quite as much to see this, you would see separate proteins here. And of course, you get the labeling of the larger proteins. And the main thing is to keep your eye on that spot. And when we've gone to ALS, that spot disappears. These spots still stay there. Now, we've done this, I think Kurt can tell you better than I can, but I know we've done it in over 20 ALS patients. And this spot has disappeared in each and every one of them. We've done it in over 30 roughly age-matched controls, and it's there in each and every one of them. We've found one person that was not diagnosed with ALS that had a protein missing in this region, and that was a person that was talked about yesterday in the first patient on, I'm blocking a name here. Stonkers. That person who was exposed to mercury and had a lot of problems had this protein missing also. So we can say that, in general, that we have a diagnostic test for ALS, and every day the numbers get better and the percentages are looking quite good, and I don't think it'll change from now. If we take CSF and run it on a single gel like we're talking about, if you remember where that protein is, this is exactly what you would see in a normal person. And we're going out with and without EDTA. It makes no difference here, but you can tell by the time we get out somewhere between 2 and 5 micromolar, this protein is selectively taken out of the system. The rest of them appear unaffected. EDTA doesn't protect, it doesn't enhance, and the same over here. And the reason it takes 2 is we find out that, depending on whether you have other metals present, you have chelation capacity left in the CSF, and it will protect you. And I would tell you that, and everyone knows this person's name that was in California that was exposed to mercury, that had Guillain-Barré disease. His CSF looked exactly like this. And that was presented to the judge, but he wasn't impressed. Now, we're going to stop talking about mercury, not totally, but we're going to go into another aspect of this because there were several people that hammered on me and pushed very hard and for us to look at the toxicity of root canal and cavitational materials. And this was something when I brought it in the laboratory, nobody wanted to do it. And Kurt will tell you that. I mean, it was passed around to about everyone because no one wanted to be involved with working with rotten teeth. And from what we do, it didn't seem very exciting, but actually it's turned out probably to be the most exciting thing in the laboratory in the next few years. And Kurt has showed this yesterday, but essentially what we were doing, we told people we would test their toxic teeth. If they want to send us some root canal teeth or avital teeth or control teeth, we would look at the toxicity testing. And to make a long story short, the test is real simple. We wash this tooth 3 times. And we use the third wash because the first wash and the second wash are too toxic. I mean, you can't tell a moderately toxic tooth from a severely toxic tooth. So This is the same tooth going through 3 washing steps, and we take the final wash, store the tooth, and we can send it to whoever you want, give it back to you or whatever, send it off for pathology with Dr. Boku, which I would recommend, and we test the toxins that are released into here, and there are very few proteins at this time. This might have quite a few proteins because the tooth is kind of bloody or has some extra proteins. But by the time we get here, we see very little protein that comes off of the tooth at this time. But we use this solution, and you understand a ml is 1,000 microliters. And in our test, we use no more, so this would be a total of 3,000 microliters total. And if you know analytical chemistry, if you wash something 3 times, you've removed 99.9% of anything that would be on the outside that would be reasonably soluble at that time. And we're coming down here, and you'll see that when I say a microliter, 1 microliter would be 1/3,000th of its total volume, or 1/1,000th of this for every microliter we talk about. And here's a screen. What this is, and I just want to point out a few things on the different teeth that we've looked at. There's a control. This is our control. This was done on brain homogeneous, so it's not like this is when we were first starting, and we were adding the extracts to human brain homogeneous. And whenever I have it underlined, those teeth are from the same patient. And where there's a question mark, they didn't tell us what tooth it was. And for example, if we look at number 67, we've got 5 teeth from this individual, and the control is right out here on the side. This is tubulin, this is creatine kinase, and this is probably glutamine synthetase, or actin at this point. And if we look at this, in his first control, which they had a thing around, it looks almost like a control, except you can see a little bit of something in this region, which is probably hemoglobin that comes into the solution. Go over to the next one, you can see you've totally wiped out tubulin. There's a little bit of creatine kinase left, but it's dramatically inhibited, say, compared to the other tooth in the mouth and to the control. And if you can go all the way across over here, here's one that's severely toxic. It took out every enzyme that photolabeled in this brain homogenate. And here's another one where creatine kinase is totally wiped out and the tubulin is not affected nearly as much, in sharp contrast to this one right here. What this tells you is that these different teeth have different microbes growing in them. They're producing different toxins and maybe combinations of toxins, but there is not a toxin. There is not a single toxin. There is a bank of them and several of them, and we don't know what they are yet. We have an idea what some of them are, but in each tooth, this could be a very complex problem, and we don't have a lot of tissue to work with. And so at any rate, we did this work, and it came out, and it was very easy to do, and quite surprising to us. And what we've seen is about 25% of root canals that we've tested show no signs of toxicity. We can't say they're toxic. 25%. That's from people who are definitely a little more on the ill side also. However, 25% of them are severely toxic. I mean, they could cause major problems. I don't think anybody would look at the level of toxicity and say this. Because if you look at a tooth that's infected, you've got 3 things that can happen. If there is an anaerobic bacteria in there, for example, you can suffer from a bacteremia because it's constantly going to be releasing that bacteria into the bloodstream, and you're going to be constantly fighting infections at other sites. Anaerobic infections, from the bacteria going in. It's going to stress your immune system because it's going to have to constantly fight this continuous release of this material into the bloodstream. The second thing is the toxins that are there. Bacteria can release 2 types of toxins. What we're talking about are metabolite toxins, hydrogen sulfide, methylthiol, and certain other metabolites that are made by bacteria, and it's their refuse. They want to get rid of it. It's the reason you have halitosis and periodontal disease. The sulfur compounds smell. Certain other bacteria release proteins, protein toxins, which are, I don't think the anaerobes that we're talking about necessarily do that, but many of them will put out proteins that affect the immune system and can affect, the adenylate cyclase system, et cetera. I mean, I would be talking about those that are much more lethal than these, such as anthrax toxin, cholera toxin, and pertussis toxin. So you have this problem. The third thing, I think, the location of the tooth and the toxin, even though it's a small amount, if someone says that's a small amount of toxin, that's not a small amount of toxin that did that. It may be a small amount, but it's a very potent toxin. And how would you like having that dropped onto your nerve cable right on your left jaw or someplace similar to that? It could cause problems, of numbness on a meridian, etc. I don't know this, but I would say anyone who said it couldn't would have a hard time defending their position if you're putting out something quite this toxic into an area where it could react with nerves in the local vicinity. To talk about these things being different, here are 2 teeth that were toxic. And both of these teeth were severely toxic when we put in 100 microliters of the toxin, or 1/10 of the last wash. But if we took this one here, which is one of the most toxic teeth we looked at, 1 microliter dropped the photolabeling of creatine kinase from that level to that level, and 5 microliters totally wiped it out. So this is a very specific toxin, and this one over here is just beginning to have a good effect out here around 30 to 50. So even in teeth that we classify as severely toxic, I guess you could say we have those that are severely toxic and we have those that are damn severely toxic, and this would be one of those. This tooth, by the way, came out of a person that was suffering from multiple sclerosis. So the teeth are all quite different. Kurt and I were working on this test, and we realized you can't use brain because we have to have a consistent marker that somebody in France or Germany could, buy the enzymes and mimic us if they wanted to test us. Because brains can be different. If you get one piece of brain from one patient and that brain's going to run out, you've got to get it from somewhere else, and it may be different. So we decided to set up a toxicity assay that would consist of us using 5 different enzymes or 5 different proteins that we knew bound ATP or would photolabel. And I would point out that Dr. Siobhan published the active site of creatine kinase, did a lot of work on that. He's also done work on AFGF. Another graduate student in my lab who's now in Sweden at the Stockholm University did the active site of creatine kinase, and we've also photolabeled these 2 enzymes. But you see, each enzyme was selected because it ran differently at a different molecular weight on this gel, so we could separate the effect of the toxins. And on this particular gel, we added increasing volumes of the toxic root canal solution and increased concentrations of hydrogen sulfide. The reason we chose hydrogen sulfide is that you know that periodontal disease bacteria, anaerobes, use use cysteine and produce hydrogen sulfide. That's the smell. And it's supposed to be toxic. It is a potent neurotoxin. Exposure to this, at very low levels will kill you. If you need the references, we have those. And here's the autoradiograph. You can see that 5 microliters of this third 1 ml wash, almost abolished the photolabeling by over 80% in each one of those for certain. And we had to go out quite a bit higher with hydrogen sulfide to get that type of effect. And I would point out here, this is 200 micromolar or 2/10 millimolar, and in certain periodontal pockets, the Swedes have reported that they've measured hydrogen sulfide concentrations as high as 2 millimolar. So that's not a little bit of toxin. But whatever's in here is much more toxic than hydrogen sulfide, and it's not hydrogen sulfide. This stinks. When Kurt and those guys would open this up, they could smell it. Hydrogen sulfide is very easy to smell. This solution, at this dilution, even though it's more toxic than this, did not have a smell that we could discern. Again, this is another one. You're going to see what I'm talking about here. This is a study using the purified proteins, and you can see our data looks a lot cleaner now, and we're getting certain types of inhibition. And again, here's a patient 104, and you look at tooth 22, 25, or 34, and you can see this tooth here, 22, which was extracted. It's a little bit different from control, slightly inhibited, but 34 out of the same mouth is a dramatically toxic tooth. And we can go over this again and again, like here's number 3. Look at that. Creatine kinase seems to be doing well, but look at the knockout of phosphoglycerate kinase and pyruvate kinase compared to the control levels. And this is the type of data we're seeing and the type of data that Effendi labeling technology sends back to you. Now, what I want to tell you, we cannot tell you what disease this represents versus this one and this one. And the reason we can't is we don't know the genetics of your patient, we don't know their age, and I'm just not smart enough to know if a tooth is located here that toxicity may cause a specific problem. I don't know the meridians, the acupuncture meridians. This is the kind of thing I would hope that as the years go on and we collect data that we'll be able to help you with. This is the, this is again comparing how toxic these things are on our purified enzyme system. This is the one that we showed a while ago that knocked out creatine kinase, etc. So we did it with the pure enzymes, with PGK and creatine kinase, increasing the toxin from 2, 5, 10, 15, 20 to 50 microliters, hydrogen sulfide from 510 going up to 1 millimolar. And as you can see, they're different. This tells you that this is not hydrogen sulfide. Our toxin at 2 microliters totally wipes this out, has no effect on phosphoglycerate mutase. However, if we go with hydrogen sulfide, when we knock out this one, after we knock out this one comes immediately. So this is much more specific for these two enzymes than is hydrogen sulfide, which seems to take them all out rather equally. So we're not talking about hydrogen sulfide when we talk about these toxins. This is a gel of cerebral spinal fluid. And if we look at this, these are the different samples, and we're going to say, is there any toxin coming out of infected root canal teeth or avital teeth that could affect the proteins in CSF? And as you can see, you can't see anything down at the bottom of this gel. There's just not enough protein there to stain effectively. This is our molecular weight markers, and each lane here represents a different CSF sample that was treated either with a root canal solution or hydrogen sulfide in micromolar concentrations. And we made an autoradiograph from this, and we see a bit more than you do otherwise. And what you see, if you remember, this is the protein that was knocked out with mercury. Look what happens with 1 microliter of this CSF, or this toxic extract, from this MS patient. That tooth is tremendously toxic. And the fact that we're not changing anything, this protein didn't change at all. This protein didn't change at all. Very selective for this protein and this protein. And you can see out here, it just wipes it out, and these proteins stay the same. Hydrogen sulfide will do the same thing, but you have to get up to near 200 to 300 micromolar to have that effect. And that shouldn't surprise you, but that's telling you that these teeth specifically contain toxins that are very selective and very potent, and mimic the conditions we see in people that are ill many times. We have a lot of research to do, but we think that this is important. I've got one slide on cavitations because they're boring. Extremely important, because I've had 5 of them removed, and I would, if I had them in again, I'd be right back tomorrow on the plane getting them taken out again. Cavitation material, we'll be happy to take your money if you want to send it to us, if you think it'll protect you from the ADA or somebody. All cavitational materials we've tested are toxic. Now, Affinity Labeling Technology would like to charge you $200,000 for each one of them and get all of them we can, but the bottom line is they're all toxic. We haven't found one yet that's not. And if they weren't toxic, they'd heal, right? So it's not a surprise. And the example is, here's a cavitational material that we collected, and we increased the concentrations. We don't know the volume of the cavitation. It's very difficult to ascertain at this time. Perhaps Bob Jones with his cavitat can go in somehow and get a good sample and tell us how much volume's there. Later on, we'll be able to tell you which ones, if they're all equally toxic or some are more toxic than the others. We don't know that, because I don't know how you guys take your samples, but you can definitely say that it's as toxic as hydrogen sulfide in this one particular solution. And this was quite a bit diluted. I mean, this was not solid. This was a liquid that we made of some cavitation material that someone had sent us. And so what we can talk about is cavitations are I can't imagine anybody saying they're not toxic and why they would exist. And definitely we've got enough data if anybody needs help in any fight about this that we can go in and say we've tested this many, we haven't found one that's not toxic yet. What we're going to talk about now, how are toxins constructed by anaerobic bacteria? Anaerobic bacteria are bacteria that live in the absence of oxygen. And if you understand, you have to have oxygen. It's an electron acceptor for you, and electron transport in the production of energy to make ATP, to make your protein, DNA, and everything else you need to live. Anaerobic bacteria, since they don't have oxygens, adapt, and they use sulfur-containing compounds that are in your body. Specifically, the amino acid cysteine and methionine are the 2 major ones. They will also use glutathione, which is there to protect you. They love it. Glutathione has a sulfhydryl on it, and it makes a good substrate for their electron transport acceptor and their modification. So, and this is a slide again made by Kurt, and it just defines the enzymes in case there's somebody in here who's a biochemist and they want to know, do we read the literature and do we know what we're talking about? This is it. There are enzymes that use this, and the reactions for making methyl-thio is shown in this slide. This is methionine, L-methionine. It's an amino acid. It's in your proteins. You eat it, you digest it, you require this. And the action of the enzyme on this is to make this alpha-ketobutyrate, release methyl-thio, and ammonia. I would point out that ammonia is one of the major activators of glutamine synthetase. It's one of the co-substrates. But methyl-thio is a unique compound. It's a toxic compound. And when we start looking at this is something that dentists should all be aware of, and medical doctors also. there was this concept, and people, it frustrated me when people would talk about amalgam tattoos. And the reason it frustrated me is I had a daughter that had one right here on her front incisor. And every time she'd smile, I'd see that purple band up around this tooth that had a root canal. There was no mercury in this tooth. She had mercury in her back fillings. We took good care of her as a kid, we thought. And I would sit at these meetings, and sometimes they're a lot worse than they are at this time, where people are showing you big open mouths that look horrible, and you'd see these amalgam tattoos, and I'd notice that some of them would be on teeth that didn't have mercury in them. The mercury would be somewhere else. So I started asking dentists, how do you see it? And they said, well, a chip of mercury flies off when they're drilling it, and it embeds in the tissue, and it turns purple. I want to tell you right now that's not true. When I looked at this, and you would say that my daughter, I don't know how she got a purple one out here. It'd be real tough to drill back here and have something ricochet off the dentist's shoulder and stick in her gum. But when we started talking about root canal toxicity, I started reading, I think the answer is right here. As you'll see it. If you have an amalgam tooth that's kicking off mercury vapor that can go to the mercury cation, and if you have an avital tooth that's doing what all avital tooths do if they're infected, producing hydrogen sulfide, mercury will come in, as a gas, will get in this vicinity, be converted to the cation, react with hydrogen sulfide, and produce mercury sulfide, which is cinnabar, which is the most stable form of mercury known, and it's the form that which we mine it out of the earth. And it collects in the tissue and forms the amalgam tattoo. And that's the reason it's such a nice, smooth, like somebody drew it with with a tattoo pen. This tells you, if you see an amalgam tattoo on a tooth, and you can go examine yourself, you can bet that that tooth, or at least, if not inside the tooth in the periodontal region, is producing hydrogen sulfide, which tells you that that tooth is infected. And if you walk around with a lot of people, you'll see them smile, and you'll see that purple amalgam tattoo on their tooth, and you can say, you have a root canal right there, and if you try this, you'll always be right. But the second thing comes down to, this is probably not that toxic, it's very stable. But if we talk about what happens if this reacts with methylthiol, we can form two compounds, methylthiol mercury, chloride, or dimethylthiol mercury, which is the dimer of this. This has to happen. If you understand mercury, vapor in your mouth, if you have periodontal disease or an infected tooth, the chemistry is real simple. It's simple. You have to form these. If you have an amalgam filling, and an infected tooth is producing these compounds, you have to produce these two compounds, and you may have to produce more. And I do believe that compounds such as this is being produced in cavitations. There's a report from Germany from a toxicologist, and cavitational mercury is extremely high in heavy metals and especially mercury. If you're producing this by a bacterial growth, you can bet that the mercury would be chelated as it goes through your gum system. It would get in there, and that's where it would precipitate, because cinnabar or mercury sulfide isn't going to go anywhere once it's formed. It's going to sit there and be extremely stable. And I don't know any way of getting this out except to mine it. All right? Okay. But remember these two compounds, and remember that we don't know everything there is to know about the type of thiol compounds that are made by these different anaerobes. As Kurt talked about yesterday, and he's done a lot of literature search. If you haven't got his literature search out there, you should look at it. There's over 300 microbes that have been identified as being found in periodontal disease or in root canal, dead root canal teeth. That's the reason you can't have a molecular biology test. You can't have an antibody test in the ELISA to test these, because there's too many anaerobes or too many microbes that can do this. So we have to look at this. But on the day that this idea dropped in my brain, and Kurt and I were talking about it, I told Kurt, go to the lab, make this. This is methylthiol, this is mercury, this is methylthiol mercury, and this is dimethylthiol mercury. And I had him make these two compounds, and the difference is this stuff does not dissolve in water. It's set there, it's biphasic. So he added the mercury chloride to this, and instantaneously it made these compounds, which did go into water. So it made them kind of more soluble in the aqueous phase. And so he made solutions of methylthiol mercury, dimethylthiol mercury, and what we're showing here is an autoreogram where we tested the toxicity. So if we just look at methylthiol, it is toxic. We just didn't go very high. This is 12 to 100 micromolar. But look at the comparison of going from 12 to 100 micromolar of the monomethylthiol mercury on these enzymes. Compare that lane to that lane. And the same thing with the dimethylthiolmercury. It's toxic, not quite as toxic as this one, and basically because you have to shed one of these groups here for the mercury to react with the protein. So the bottom line is these compounds appear to be very toxic to mammalian enzymes or human enzymes. If we take them in cell culture and we look at the proliferation of HL60 cells in culture and we run the concentration, this represents 100% killing up here. We're going from 0.01 micromolar at 1 micromolar, and at about 0.8 we see this starting to kill. We're comparing it to mercury, and by the time we get out to about, I'd say 3 to 4 micromolar, we've killed all the cells in a short culture with these compounds. Many of you read the story about the Dartmouth University professor who was a lady who smelled 2 drops of dimethylmercury on her glove and subsequently lost her hearing, her sight, went into a coma, and died 10 months later. If you look at this compound, it's dimethylthiolmercury. It would have the same properties, and you've got to understand the difference between chemical reactivity and toxicity. Many compounds, and if we look here at mercury, mercury Hg2+ is more reactive than methylmercury and dimethylmercury. It's more reactive. The thing is, it's also easy to pick up and protect yourself against. If you're going to get the dimethylmercury to have a reaction, it has to lose one of those methyl groups. But in the process, while it's being exposed, it hits into the lipid layer or the hydrophobic layers of the body, getting away from glutathione, metallothionein, which are water-soluble proteins, and it can spread throughout the body, get into the mitochondria, etc., and in a way, it's a slower, more agonizing death. I think that dimethylthiolmercury and methylthiolmercury are 2 of the toxic compounds that are probably responsible for people and this tight correlation we see between periodontal disease and stroke, cardiovascular disease, atherosclerosis, low birth weight babies, etc. These compounds, even though there's very little probably being produced in these mouths, is enough to severely affect your health. It has to be. Yes?
The lady at Dartmouth College that had dimethylmercury on her glove and within less than a year she was dead.
10 months.
10 months.
Yes. And she was in a coma in 3 months and could not walk or work within a week. But it took her a long time to die. So this is an insidious poison. And you see the chelators that we have today, DMPS and DMSA, couldn't help her because this is locked in your lipid layer, and DMPS and DMSA are both charged compounds. They won't go into the lipid bilayer. So now we're going to talk about just something. This is the curvicular fluid test which Kurt Pendergrass has developed. And what it's come out to be better than we ever imagined. And so what we're going to talk about just for a few minutes to let you know that we are constantly in a state of flux. Every time we think we've got this worked out, we find something that will improve it. And if you listened to Kurt talk yesterday, you've heard some of this. But I want to point out we have yellow arrows here. And these arrows are pointing out certain things. And this is the stain gel. Here are our test proteins, one of them with a washed pick and one without a washed pick. All this is to tell you is that if you try to use an unwashed pick, we can't do the sample for you because it's going to inhibit it. And you'll see that on the autoradiogram. But you can see the proteins that are here are all equivalent and look alike. And in many of the controls, what we see across here is a little bit of albumin that comes out, with the exception of this one. And I have no idea what's there, except that is not a control tooth. That is a toxic tooth. There's 2 of these. This one and this one are turned around. This should be a root canal, and this should be a control. But if we look through here, on the staining, there's no protein here. If we get here, here's a protein. And this protein appears in 5 of these samples. And this protein is in the curvicular fluid, and it's not supposed to be. On most of the controls, you'll notice that it's not there. Here's another exception. We see this just once in a while, not too often, but we do see this one, and this is a very toxic tooth, as we'll show later on. Over here, you can't see anything, but if you look down through here, you'll see that some of these proteins are breaking into 2 bands, and you'll see that there's another protein on photolabeling that sticks out like a sore thumb in this region. So if we take a look at this, I want to make one more point. This is albumin. And what we see in many of the bacterial infected teeth, that the albumin level goes up in the curvicular fluid. Probably a protective device by the body. So this is probably from the human. This may or may not be from the human. The one we're seeing down here is definitely from the bacteria. This one we think is from some other microorganism, likely not a bacteria, but perhaps a yeast, a fungus, or an anaerobe. An amoeba.
Boyd,
Yes?
do you have any information at all, is the toxicity of these substances due in any part to the immune system response to the substances produced by the gram-negative pathway?
We haven't studied that yet. That would really be a good study, but that would take a lot of money. I mean, that would take me collaborating with somebody and I have to pay them. This I steal from other grants and do. Okay. But if you remember, these arrows are in the same place. And if you look at this, here's our control. First thing we can say, if you don't wash it, if you don't use the picks that Kurt prepares for you, you're going to get this kind of data. But you notice, this affects the photoprobe because everything is dropped off equally. There's nothing selectively wiped out like you see in some of these others where one protein will be missing like this one, and this one is still there. But take a look at this. If you look at this particular one, this is one where I showed you where we couldn't see a protein. Here's that extra photolabeled band, and this is the molecular weight of bacteria alkaline phosphatase. You can't see it on the gel, and it's very difficult to measure it, and it's not there in a lot of them, but what we can do is it binds phosphates. It takes phosphates off a thing. That means it binds our probe, and so we label it. The same thing up here. We think this is another form of an alkaline phosphatase, and you can see that wherever it is, there's also a protein band that runs above it, because this should run right in here. And you can see this is very high in several of these samples. And once we identify totally what this is for certain, we'll be able to tell you what kind of infection is in that particular tooth. And you can see it's only in 5 of them. Over here where we found that usual protein that's set down there, look how toxic that tooth is. There's no alkaline phosphatase at either level. Just an extremely toxic tooth. I mean, this is probably the most toxic on the page here. So what we're seeing then with this photolabeling technique is that we can measure, by the way, using a computer off of this instrument. We just draw an aerial around this, just come in and circle this, and it'll tell us how many counts are in that band, and we can compare it all to the control bands. And for example, if you look here, if you go through here, these two aren't going to be very much inhibited in this regard, but this one's going to be wiped out, and look at this big band jumping up. So we see the test enzyme dropping down, this one dropped down also, and here's a big one spot lighting up. So we have a test, a double test that tells us there's something in there that shouldn't be there, and it's killing our test enzymes. So this is going to turn into a slick test. Now, the drawback of this is creatine kinase, and we're having other enzymes. We know this is much higher than this. There's a phosphatase similar to this one that's co-migrating with our creatine kinase and giving us abnormal readings. So we're going to change our gel procedure or the proteins we're using, and we're going to separate this out. So this is the kind of refinement. We have to go through a refinement every time we run into a problem. But this is going to work. We can see that now. It's working very well today because you can compare it. I mean, even though we can't tell you how much is here, we can sure tell you that something bad happened right there, and it didn't happen in these 2 teeth. So the conclusions of all this is, the chemicals found in cavitational materials are all toxic. Root canals are toxic, not all of them. I know a lot of people get upset at me because they think Weston Price was right and he found toxicity in every tooth. Well, we're measuring our type of toxicity. That doesn't mean he was wrong or we're wrong. Just with our test, we see that 25% of the teeth are not toxic. They're not putting out toxic metabolites. My opinion about root canals is they're sort of like a tire you put on your car. You have to check them every month, and sooner or later they're going to go bad. And you're going to have to replace them or do something with them. For certain, we have a test where we can show this. And if you realize, we're doing this on 6 or 7 microliters of curvicular fluid, and you saw the amount of albumin we're getting out, so there's no damn doubt we're getting something in there. We're getting a good sample. And if you send us saliva, we can tell the difference because the proteins in saliva have a different staining profile than the proteins in curvicular fluid. So these things work. There are certain things that do bother us. If somebody gargles with Listerine and swishes it in their mouth for 10 minutes and then does our test, we may get a weird result. If you have them chew on these tablets that you like to have them chew on to turn their mouth purple and green, and we know some of you do that because we see it when you send it to us. We can do spectra. We've gotten samples that are green and purple. I think, was it purple? Green and some color, I know that. But at any rate, we can measure the toxicity, and the technology allows you for the selective toxicity of against a bank of several purified enzymes, but we cannot tell you when we see severe toxicity or toxicity against one enzyme what disease that will cause. because we don't know the patient, we don't know their age, we don't know their sex, we don't know any other problems they have, and we haven't done enough samples yet. Perhaps someday in the future, because we're putting all of this data on a computer, we can go through and pick out a particular protein that shows up, take all the patients from that and see how many of them had a particular problem. But we haven't got the numbers to do that today. We've done well over 700 teeth or ventricular fluids now. Terry?
You don't know how long the root canal's been in the mouth either, do you?
We know nothing. I mean, you can test it. You can trick us if you want. You can send us all kinds of crap, but we'll let you know you did it. I have one more slide. This all it says is, please be aware that any silver lining found within me may contain trace amounts of mercury. And I think that's a good. And this is copyright from David Farley, a cartoon that my technician made for me. Thank you.